Okay, now we want to solve this March 2023 exam. You all have it in front of you? Okay, get out a paper. I'm going to start first by the questions that I thought were a little bit tricky, and then we will finish the remaining exam. Okay, I want you to open that question first. Yes, the question on page 4 of the exam. Question on page 4. The question here, B. You all have the question in front of you? Figure 1.2 shows the pressure of blood flowing through different blood vessels as it travels around the body. Venules are narrow vessels that connect capillaries to veins. On the x-axis, you have the type of the blood vessel, and on the y-axis, you have the blood pressure. Now you have arteries, and then followed by a smaller blood vessel, capillaries, venules, and veins. What is X? Arteries give what? What's after arteries? Excuse me? Correct. Would you have known the answer on your own? Why? Didn't we take the capillary bed and we took that the arteries break down to give me the smaller arterioles? And then you have a capillary bed, and then they give me the venules and then the veins. Yes, we took this. And the pressure is highest always at the arteries, and it is lowest at the veins, not the capillaries. At the capillaries, you have a little bit more pressure than the veins. So X are the arterioles. Arteries, they break down into smaller arteries to give arterioles. Clear? Now, state the name of the blood vessel that is represented by X. X is the arteriole. Explain reasons for the changes in pressure seen in the arteries. Okay, I want you to go to the graph. Here are the arteries, and the pressure, as you can see, looks like this. We want to describe this, and we want to explain it. How will you describe the graph moving up and down, up and down, up and down? Excuse me? Very good. This is a very good word. You will say that the pressure what? Fluctuates. Or you can simply say that the pressure goes up and down. Now, this is the describing. Now, explain. Why does the pressure here in the arteries go up and down? Yes. You are right, because the heart is contracting and relaxing, okay? Because of it goes up when the heart contracts, the pressure will increase. And it goes down when the heart relaxes, the pressure will decrease. Now, if you look at the mark scheme, this is exactly the answer with an added word. There is only one word, because what is the tissue in the body? The only tissue in the body that contracts and relaxes, bones or fats or muscles? Muscles. This is the word that they wanted, that the heart muscle will contract, the pressure will increase, or the heart muscle will relax, the pressure will decrease. And remind me, what is the name of the muscle in the heart? Cardiac muscle. So cardiac muscle will be a very good term. Cardiac muscle will contract in a heartbeat, the pressure will increase. Cardiac muscle will relax, the pressure will decrease. That is why the pressure is going up and down, or it is fluctuating. Okay? Now, this was a question. The next question, explain the structural adaptations of arteries and veins. This is a very direct question. I want to solve the indirect questions first. Good. Now, question number 3 on this exam, page 9. Because you get to ignore the questions that we took at the very beginning of the year, classification and cell structure. And this question here is over classification of plants. Figure 3.1 shows part of the classification for plants. Now, complete figure 3.1. Plants are divided into the ferns and the what? What are the ferns? Remind me. Huh? Yes, flowering or non-flowering? Non-flowering. We took it at the very beginning of the year, and if you decided to ignore these lessons and you stepped into this exam, you will not be able to solve this question. Okay? So the ferns are the non-flowering plants. So what will be the answer here? Flowering. Very good. And the flowering plants are divided into the monocot and the dicot. Excellent. Again, it's a straightforward question, but I assume that you will ignore those lessons. Describe two features of monocots. No, they have flower parts in multiples of 3. And talk to me about the leaves. The leaves are broad or thin? Thin. They have long, thin leaves. And what about the veins? No, parallel. Parallel veins, straight veins. You just forgot. Okay? So the leaves are long and thin, and they have parallel veins, and they have flower parts in multiples of 3. How many cotyledons do they have? The first, yes, 1 cotyledon. So let's write all of them. 1 cotyledon. Leaves are long and thin. Veins parallel. And the flower parts in multiples of 3. Okay? Another tricky question in this lesson, in this exam, is the next one, B. It's easy, but let's see if you will remember. No. Plant reproduction. Figure 3.2 shows the life cycle of a fern. The life cycle of a fern has some similarities and some differences compared with the life cycles for other plants. The letters represent processes that occur during the life cycle, haploid stage of the life represented by N, and the diploid represented by 2N. Okay, you had here an adult sporophyte, 2N. 2N is the number of what? It is representing number of what? Yes, diploid, very good. So 2N is a diploid cell or the number of chromosomes, yeah? And then N, what is A? Yes, N is haploid. What is the process A that changed diploid to haploid? Correct. A is meiosis, of course. What is B? You had actually B, adult gametophyte, they just made more. They reproduced more. They will not ask you about B. What is D? They will ask you about D. Fertilization. B is multiplication. Those spores, they multiply by mitosis, okay, and then they fuse in fertilization D to give me a zygote. State the number of chromosomes in the body cells of the gametophyte. If the adult sporophyte has 1200 chromosomes, the gamete, yes, 1200 divided by 2, this will be 600. You all know how we got it? Yeah, divided by 2, okay, because this is diploid and this is haploid. Identify and describe process A. What is process A? Meiosis. Now, remind me, what is meiosis? I want at least 3 sentences to describe meiosis. Excellent. Thank you so much. Excuse me? I can't hear you. Meiosis? This is here, you are describing sexual reproduction. Yes, meiosis happens in sexual reproduction, but this is the division that happens inside the cells. So meiosis is defined by reduction division. Reduction division. Some of you will tell me nuclear division. It is nuclear division, of course, it happens in the nucleus. Both meiosis and mitosis are nuclear division, but meiosis is specifically defined as reduction division. Why? Because we start off with a diploid cell and we end with a haploid, okay? And these haploid are genetically identical or not identical? Not identical. You will write all that and you will define what are haploid. So meiosis is reduction division that produces genetically non-identical haploid cells. Is this enough? No. What will I do next? Exactly. Excuse me? You can say that, of course. But when you mention haploid cells, you will leave it or you will define it? Huh? You will leave, if you mention the term haploid cells, you will, of course, define it. It's 3 marks. Okay? You will define what are haploid cells. What are they? What's your name? Yes. What are haploid cells? Yes, yes. No. You're right, but this is not the definition. One set of unpaired chromosomes. Okay? Give me examples. Examples in your body of haploid cells. You only have 2 haploid cells. Haploid, not diploid. What is the cell that is haploid in your reproductive organs? Egg. Egg or sperm. Okay? Any other cell in the body is diploid. Even the zygote is diploid. Okay? So you will define what are haploid cells. The haploid cells, these are cells with one set of unpaired chromosomes. Example, sperm/egg. In the plant, it's called pollen and ovule. No sperm and no egg in plant, okay? And you can also, if these are 2 sentences and you want to write a 3rd sentence, you can say it happens in sexual reproduction only. You can say that it happens in the reproductive organs only. You can say that we start with a diploid cell. We start with a diploid cell and it is converted into a haploid cell. Diploid is represented by 2N. Haploid is represented by 1N. And you have 4 rounds of division. Remember the table? You have 2 rounds of division, and you end up with 4 haploid cells. You can write everything you know about meiosis, but the most important thing is that you have to write the definition, which is reduction division. Reduction division. Mitosis is divided as nuclear division that produces genetically the same cells or genetically identical cells. Yes. What's this? You'll do what with it? Wait. Let's do the important stuff now. Okay, what is process D? Fertilization. What is the definition of fertilization? Remind me. What, excuse me? Fusion, not fusion of zygote, fusion of the nuclei. Fusion of the male and female nuclei. And don't say fusion of cells. It's fusion of the male and the female nuclei. Please remind me. You have all those definitions. Remind me to prepare a year 10 definition page with most of the definitions are in reproduction and inheritance. Okay, this is a direct question. We will solve the indirect questions first, like this one on page 12 of this exam, number 4. Now look at this question. For figure 4.1 is a photograph of a tree that has been girdled. Girdling involves removing a complete circle of bark and phloem from around the tree. So here they removed the external layer, which is what, the xylem or the phloem? Phloem. They told you in the question they removed the phloem. Now this question out of 4 marks. Explain why the area above the girdle in figure 4.1 will become swollen and use the terms source and sink in your answer. 4 marks. Please focus before you answer because you said one big mistake. Phloem transports minerals, sucrose and amino acids, not glucose. Source to sink. And this process is called what? What is the function of the translocation? You have an exam in a couple of days. Yeah. Of course. Yes, you can. Yes. Okay. Now, they mentioned the word phloem and they told you use the terms source and sink. So I have a lot of things to say. So I will start with the function of the phloem. What is the function of the phloem? Very good. So the phloem, okay. Function of phloem. Translocation, and then define it. I cannot write the whole thing here, which is the transport of sucrose, it's not glucose, sucrose, and amino acids from source to sink. You mentioned source and sink, define them. Okay, this is the art of answering an essay question. Nice, clear, in order. What is the source? Yes, this is an example. The definition of the source. It's the plant part that releases sucrose and amino acids. Example, leaf. Okay, you have a problem with essay question. Source are the plant parts that produce or slash released sucrose and amino acids. Example, leaves, and not even any leaves. Adult photosynthesizing leaves, adult leaves that do photosynthesis, because baby leaves still don't photosynthesize. Adult photosynthesizing leaves. What is the sink? What is the sink? Define the sink. What's the sink? Excuse me? Very good. It's the plant part that will receive or store the sucrose and amino acids. Example, roots or storage organs. So the sink I have written down, these are the plant parts that receive or slash store sucrose and amino acids. Example, storage organs, like a potato. Example, roots. Okay? Now, we still did not finish answering the question. Why does this part above the part that has been cut, why does it become swollen? What's your name? Yes. Yassine, my dear. Now we mentioned the function of the phloem and the source and the sink, and we gave examples, and we are amazing. Why would this part become swollen? Very good, because translocation will stop, because the sucrose and amino acid will not be able to move from the source, which is the leaf at the top, to the roots at the bottom. So it will accumulate here, and it will make it swell. Okay? So here you introduced translocation, source, sink, you gave examples, and then you gave them their answer. It will swell because no translocation will happen. We remove the phloem. So the sucrose and amino acid will not be able to move from the leaves to the roots. So it will accumulate and it will make it swollen. What's your name? Habiba, you understand the question? Please highlight this question. In every exam, there is a question that is not very nice. So highlight those questions when you find them, so we go back and revise them. Good? No, start from the recent exams, 2023 backwards. So start from the recent, the most recent. So again, this, in my opinion, is a question. Can you solve it like this on your own without help? On your own without help, can you solve it like this? Look, if you have all this space, for example, please don't give me a messy paragraph, and also don't do bullet points. Some of you, they like doing bullet points and it helps them remember. It's okay. Do the point or do the bullet point on the line itself. Like, don't make a clear star. You can do this on the line itself and write your sentence. And please write one sentence per line. It looks very neat and it will organize your thoughts. Okay? And you can do it this way. Source, 2 dots, and write the definition. Sink, 2 dots, and write the definition. This messy answer of yours is horrible. Horrible. Fine? Let me show you an example of a messy answer. I think I sent you once one messy answer that was... no, I don't have it here, unfortunately. Okay. Now, another question that I thought was not easy. All the remaining questions are direct and straightforward. We're actually done. These were the questions that were not easy. Yes, this one. An experimental question. You can find experimental questions in paper 6 and paper 4. This question here is very easy. Movement in and out of cells. Habiba, pay attention. Answer me. Help me, because I don't know. Figure 2.1 shows the apparatus. Here you have starch, only starch, no enzyme. So here in this whisking tubing, what's a whisking tubing? Remind me. Partially permeable. Artificial membrane that is exactly like a semi-permeable membrane. Partially permeable. Here in this whisking tubing, I have only starch. Okay, Habiba? In the other test tube, I have starch and the enzyme amylase. And then they said in the question that The liquid outside the dialysis tubing in test tube A and B was tested with Benedict at 0 minutes and after 30 minutes. Table 2.1 shows the results. After 30 minutes, test tube A remained blue. And after 30 minutes, test tube B changed from blue to red. Can you tell me, Habiba, what happened so that test tube B will give me this result with Benedict? Yes. Why was there a color change? What did the amylase enzyme do? Which reaction? Yes, of course. Here you have starch and no enzyme. Here you have the starch and you have an enzyme. This enzyme will break down the starch into what? No. Maltose. Maltase will break down maltose to glucose. Amylase will break down starch to maltose, which is the reducing sugar, which is going to give me a positive result with Benedict. Okay, Benedict is not only for glucose, it's for any simple sugar, any reducing sugar. Maltose is broken down by maltase to give me glucose. So it doesn't break down to glucose, it breaks down to maltose. Okay, and then when the maltose is broken down, what will happen to this maltose? Why does it give me a positive result in the external water? Because we took water from outside the whisking tubing. Exactly. The maltose is small. It will diffuse from the inside of the tube to the outside by diffusion and mention the definition. And that is why when we took a sample of this external water and we tested this sample with Benedict, it will give me a blue color. Here, the starch is not broken down and it's big. It will not move out of the whisking tubing to the outside. And even if it does, it will not give me a positive result with Benedict. Diffusion, of course. Of course, diffusion. This is non-living. Active transport needs a living cell membrane. Okay, so this question, also mark it. This is a question that is critical thinking. I assume you will do horrible mistakes. So this question, out of 6 marks, oh my goodness, 6 marks. You will talk about test tube A and you will talk about test tube B. And you will talk about how the maltose will move out, which process, diffusion, and define it. And it's easy. Just understand the question, read the questions carefully. So in A, you will say that the starch, is it broken down or not broken down? Not broken down. Because there is no enzyme. And it is too big. It will not diffuse out of the whisking tubing. This is all what you can say in A. That is why it will give you a negative result. But B, you will say a lot of things. Can someone repeat again what happens in B? Very good. The starch will be broken down by the help of the amylase enzyme to give me maltose. And this maltose will then diffuse. It's small in size, okay? It will diffuse out of the tubing. You have the exam? Out of the tube and go to the external water. And this will give me a positive result with Benedict. I will not end here. I have to define what is diffusion. What is the diffusion? Who will remind me? Yes, it's the net movement of molecules down a concentration gradient. No energy is needed. No cell membrane is needed. No membrane proteins are needed. That's it. And you will take the 6 marks, I promise. Okay? And you can also write the, Yes, of course. Okay? So here 6 marks because you have to discuss what happened in A and what happened in B and mention everything about diffusion and define it, etc., etc. And say what is the whisking tubing. It is an artificial tube that is semi-permeable. 6 marks, so write the whole lesson. Good? This was also a question that I assumed was not very easy. The remaining questions, the remaining exam is direct, straightforward, and depends directly on how much you studied. And not a single question on this exam is core. All of it is supplement. That's why I tell you, swallow year 10 notes. Year 10 notes are enough to make you get an A, a 9. Because paper 4 is all year 10, and paper 6 is a different story. Paper 2 is a mix, but paper 2 also, most of it is supplement. So study 10 very, very, very well, and then go back to 9. Good? Let's solve the whole exam and finish it together. And we will do inheritance online, yeah. Okay. Can you please sit here so you see the exam? Okay, figure 1A is a simplified diagram of the circulatory system. State the letter that represents the blood that contains blood with the highest concentration of oxygen. Choose here one blood vessel that has the highest oxygen. A is wrong.
B.
Yes. Look at B. B here, the blood is coming out of the lungs. A is what? Yes. Can you come here? A is what? The aorta. And the aorta is oxygenated or deoxygenated? Oxygenated. Okay, you have the option to choose A and B. Which one will you choose? They are both oxygenated, by the way. B. Because B is coming directly from the lungs. You see? Okay, so the answer is B. State the letter from figure 1.1 that represents the hepatic portal vein. Where is the hepatic portal vein? Yes, this is the vein that carries the digested food from the small intestine to the liver. So it is C. Clear? State all the letters that represent arteries. Arteries are blood vessels that go out of the heart or towards the heart. Out of the heart towards the organ. So choose any letter that is moving towards an organ. A. Y is what? Y is the septum. A or E. Yes, E is an artery. They did not say oxygenated or D. And D. Yes. That represent arteries. Yes. So it is A and E and D. Fine? There is no letter. This is a vein. Okay. Explain the importance of Y. What is Y? Very good. What is the importance of the septum? Very good. Do I have to write it down for you? Right. It provides a double or helps for a double circulation. Excuse me? So this is the septum. It prevents the mixture. I will not write it down. Prevents mixing oxygenated and deoxygenated blood. And it helps in providing a double circulation. Okay. State the evidence. Yes, state the evidence from figure 1.1 that shows that this is a double circulation. Okay, this is the definition. You will say that the heart will go through the heart twice per circuit. That's it. What are the two circulations? Yes, what are their names? Yes, and no, this is a double circulation. What is the definition and what are the two circulations? You told me that the double circulation is when the heart passes through the heart how many times per circuit? 2 times. What are the two circuits in a double circulation? What? Very good. One to the lungs and one to the rest of the body. The one going to the lungs, what is it called? Yes, yes. Okay, you have the pulmonary circulation, the one going to the lungs, and you have the one going to the rest of the body, which is the systemic circulation. Which one is the high pressure and the low pressure circulation? Let him answer. The low pressure circulation is the pulmonary or the systemic? Low pressure, low pressure. I'm moving slowly because I don't want the blood vessels to rupture. Pulmonary, and I don't want the delicate alveoli to rupture. So the pulmonary is the low pressure and the systemic is the high pressure. I want the blood to be pumped very quickly and very strongly to the whole body so it reaches quickly. Okay, Habiba, are you with me? Fine. So again, anything critical? No. These questions are all straightforward and all supplement. This is not core. Fine. We answered this question. Here, using the information, explain the adaptations of arteries and veins. Straight, direct question. You want to make a line here in the middle of the whatever and write arteries and veins. And I want you to tell me what are the differences. Narrow lumen, very good. Why? Yes, to withstand blood at high pressure. What else? Again, very thick walls. But these thick walls, it's not enough to say that they're thick. What do they contain in those walls? Which tissues? Which? Muscular, yes, they have muscles, thick muscles, elastic fibers, collagen. I think I mentioned that. Okay, they have lots of elastic fibers to make them elastic so that they can stretch and recoil and stretch and recoil. So I have very thick walls. And the veins, compare the walls of the veins, thicker or thinner? Thinner. Thinner walls, yes. And they have a wide lumen or narrow lumen? Wide, because they carry blood at low pressure. Okay. What else? I want a third difference at least. Very good oxygenated blood, except there's one exception. Yes. And what else? Arteries. Do they have valves? No valves. No valves and arteries. Do they have a pulse? No. Yes, arteries, they have a pulse because of the high pressures. They have elastic, the thick walls don't just say thick walls. They have more elastic fibers, more smooth muscles. Okay? Now, and the veins are the exact opposite. The veins, wide lumen, they carry more blood, low pressure, less resistance, arteries, everything is the opposite. We answered this question. This is 6 marks, 6 marks. Who thought of active transport? You would write active transport here, I'll kill you. Active transport needs a living cell membrane. Okay, now this is so easy. The pepsin enzyme, what does it work on and what does it produce? Of course, proteins into amino acids. And the fatty acid and glycerol, what is the substrate, Habiba? What is the enzyme that breaks down a substrate that will give me fatty acid and glycerol? What's the name of the enzyme? Yes, lipase. Of course. What does it break down? Fats into? Yes, trypsin. Again, they're trying to confuse you. Exactly like pepsin. Exactly. It breaks down proteins into amino acids. But where is pepsin found? Very good. Trypsin. Small intestine. Glucose. What will break down what into glucose? Maltase, not amylase. Maltase is the enzyme. It will break down maltose into glucose. Again, this is supplement. Why? Because this enzyme is not in the core and the trypsin enzyme is not in the core. Okay? State the name of the structures that increase the surface area for the absorption in the small intestine. Very easy. We answered this one. And we answered this one. Very good. What's the name of process D? Fertilization. Figure 3.3 shows the part of 2 flowers from 2 different plants. What is species? 2 marks. Again, this is classification. The very first question, the lesson in the syllabus. Organisms that can what? Very good. They can reproduce. Not only that, they can produce fertile offspring. Okay? So these are organisms that can reproduce. And by the way, they're similar. They have similar structure, similar morphology, similar behavior that can reproduce and produce fertile, the offspring, they have to be fertile. They have the same morphology and internal anatomy. Not only that, they have to produce and bring fertile offspring. Again, this is supplement. Or maybe this is the only core question. Annotate figure 3.3 to show the mechanism of cross-pollination by labeling the structures involved and drawing an arrow to show the pathway of the pollen. Okay, what's your name? Yassine. What is, can you draw here cross-pollination? Very good. The anther produces the what? Very good. So I will do this. This is my anther. Okay. And I can write between brackets pollen. And this anther or the pollen will move from where to where? To the stigma of another flower or the same flower? To the other flower. This is cross-pollination. So you will do this. I don't have here. Oh, it doesn't work. This is the stigma. Okay, that's it. This is cross-pollination. This is what you will do on the diagram. Clear? Okay, nice. Explain the disadvantages of cross-pollination compared with self-pollination. Direct question. Huh? We did? No, we didn't. Oh, yes. Draw an X to show where the fertilization happens. Where does it happen? Yes, here. Here, it doesn't, right. Okay. Explain the disadvantages of cross-pollination compared with self-pollination. Remember the table? Cross-pollination produces more or less variation? More, but they don't want advantages, they want disadvantages. Which are the disadvantages of any type of sexual reproduction? Yes, cross-pollination. I'll give you the answer now because I have a B here. They want the disadvantages of cross-pollination. Okay? So this is an example of sexual reproduction. They cannot produce in isolation because they need pollinators. And it takes more time or less time? More time, more energy, more time. Give me another disadvantage. Slower or faster? Slow. Needs more energy or less energy? More energy and more pollinators. Now, what is the remaining part of the question? Compare what? Disadvantages of self? Okay, so you will write this part and you will write this part. Okay, not only the disadvantages of cross-pollination, also the disadvantages of sexual reproduction, because it is sexual reproduction. Fine, so the disadvantages, write it down, don't look at me. It needs pollinators, it cannot reproduce in isolation, pollen will be wasted, and this is sexual. I will also need more time and I will need more energy because this is sexual, okay? And the disadvantages of the sexual, of course, the list of no variation. No variation. It cannot be adapted to changes in environment. It will die if there is a disease. There is a higher chance to become extinct. It will not show mutation. It will not allow natural selection. Another disadvantage of sexual reproduction is that if there is an unfavorable characteristic, it can pass on. Genetic diseases can pass on. So you have a lot of things to say. This is the direct question. It directly depends on how much you studied. You studied, you will answer. You will not study, you will not answer. Again, this is supplement. Why are you looking at me? Write the answer. Clear? Done? Okay, we answered this question. This was a tricky one. Explain why the leaves will still be able to receive mineral ions from the roots. Yes, the xylem was not removed. And the xylem, its function is to transport water and mineral ions. Yes, from the roots to the leaf. Conduction and transpiration stream. So the xylem here is not removed, it's intact. Function of the xylem, conduction. You write it down, please, which is the transport of water and ions from the roots to the leaves in the transpiration stream. up in one direction. They always like the term transpiration stream. Transpiration stream is the movement of the water inside the xylem, not its evaporation in the leaves. No, they don't need. Explain why glucose made during photosynthesis is required for the absorption of mineral ions by the roots. Now again, if you understand the question, you will understand how easy it is. How are the ions absorbed by the roots, Habiba? Habiba, how are the ions absorbed by the roots? Which method? Very good. So you will mention everything about active transport. Down a concentration gradient, needs energy for the change of shape, for the carrier proteins and a living cell membrane, from respiration, write it all. Okay, so you write here everything about active transport. Against a concentration gradient, excuse me? I will not get anything. I will not make an exam. I will choose an exam. So I'm going to simply choose an exam, just like I did in paper 6. Did I play around with the exam? No. But it has ecology, I promise you. A lot. Actually, after I sent it, I realized that it's a lot of ecology. That's why I'm telling you, study ecology, study ecology. It's not hard. I told you study year 10 notes only, which are 9 pages. 9 pages. I told you leave year 9 ecology because it's repeated in year 10 with the additions that you need. So there are 6 pages and 3. Very easy. And even for Cambridge, trust me, study year 10 ecology. It's enough. It's the same. Eutrophication and deforestation, these are the main points. Global warming, they're found also in year 10. And the conservation and taking care of the endangered species, advantages and this, and what do you do, and this list. That's it. Okay? What's the balance symbol for photosynthesis? Again, direct question. Photosynthesis. Excuse me? Yes, 6 CO2, carbon dioxide, and water. Yes. In the presence of light, write light here. C6, chloroplast, H12O6 plus. Yes, correct. Again, this is supplement. So paper 4 is all supplement. And on the night of the exam, because I will be bombarded with questions, I know, only study the supplement. Fine. Explain why the eye can be described as a sense organ. Excuse me? Yes, this is 2 marks. A sense organ is an organ that has lots of receptors. And continue the definition. What do these receptors do? Yes, they detect and respond to a specific stimuli. Like the eye, what is the stimulus that they respond to? Light. Okay, they have light sensitive cells. Write it down. I don't have to write it down. Okay, this is the mark scheme. I sent you even the mark scheme. Receptor cells that detect and respond to specific stimuli. If you want to give an example, give an example. This is here, we're number 5. What are the 2 parts of the central nervous system? Brain and spinal cord. Okay, let's label this. Yes, there is a question here that's not nice. I want to answer it. Here. E. Explain why when a person is unable to focus on distant objects, if the suspensory ligaments becomes permanently overstretched. This question is just twisted. What happens? You know what this question is? What happens when you look at a distant object? What happens to the lens? How? 1 minute. What did you say? What did you say? Yes, this is lens accommodation. What happens when you look at a distant object? Yes, you will mention that and you will say that this process, will not work properly. It will be overstretched and things will not happen properly. Yes, they twist the questions. They want to confuse you. This is the intention. Look, this is accommodation. Yeah, if you forgot, you have here your lens. It is held by the ciliary muscle and suspensory ligaments. When you look at a far away object, we said we're sitting on the sea and relaxed. What will happen to the ciliary muscle? Far away, distant. I'm happy and relaxed. I'm in the North Coast. Relax. When you're studying and you're looking at your phone and it's very close, this is when your eyes are contracted. When you are sitting on the beach, this is when your ciliary muscles and your eye muscles relax. So the ciliary muscle will relax and the suspensory ligament will become tight. Yes? When this happens, how will this affect the lens? Thin? Very good, it will be thin. And then less refraction of light or more refraction? Thin. Less refractive power. Okay? So less refraction. And the light will hopefully fall on the retina or fovea. Yes. When you have something wrong, this doesn't work. So when the suspensory ligaments are overstretched, the accommodation will be faulty and the lens will not be thin. The lens will actually be thicker. They are overstretched, they're loose. So when I come to stretch them, they will still be loose. Okay, so this is not going to operate properly, and the lens, instead of being thin, it will be thicker. The opposite will happen. Look at the answer. Here is the answer. See? Any three from ciliary muscle will relax. Suspensory ligaments can no longer be taut. The lens is no longer stretched. It will remain wide. Angle of refraction remain unchanged. So in other words, you will mention what normally happens and you will say that it will become faulty. See? So again, the question is twisted. But in the end, they just don't want to ask you a direct question. What happens in the lens when you look at a distant object? So they will change the question, but the answers are the same. I keep on telling you that. They play around with the questions. Fine? Clear? So you will mention what normally happens and you will say that this will normally not happen because the suspensory ligaments are overstretched and the lens will be normally thicker than normal and it will not fall on the retina and you will have a blurred vision. You will not see properly, okay? Omar, where's the gum? Oh yeah, fine. Good. So here, continue this question. Draw X on the figure to identify the blind spot. Where's the blind spot? Right here. Exactly in front of the optic nerve. Okay. Can you do this alone? What is H? Where is H? Yes, the retina. And what is found here? What is the function of the retina? It has the light sensitive cells to detect light. Excuse me? The rods and the cones. Yeah. What transmits impulses to the central nervous system? Optic nerve, which is which letter? G. Good. And then cornea, what's its function? Refraction of the light as the light enters. And which letter is it? No, C is the pupil. D. Okay? So write it down. If you notice, the remaining questions are straightforward. There were around 2 or 3 questions that were a little bit tricky. These questions are what makes the difference between a 7 or an 8. Done? Genetic engineering. What is B? Yes? B is the gene that I isolated. What is this? The plasmid. What is this? Yes, the recombinant. When I fuse the plasmid with the gene, this is the recombinant. Write it down. Very good. And then here, what happened? Which type of reproduction happens here? Tissue culture. Sexual or asexual? Asexual, of course. Good. Continue this on your own and tell me. The part labeled A is called what? What's A? Plasmid. We said it, okay? It's called the plasmid. Yes, Yusuf? Yes, see. Part A is extracted and cut using what? What is the name of the enzyme? Restriction enzyme, forming what? Sticky ends, okay? Because I don't know why you don't believe me when I tell you that you need to know the name of the enzyme. So these are restriction enzymes to give me sticky ends. The part labeled B is the part that codes for the production of the beta carotene. Yes, it's a gene. And the enzyme used to cut part A is also used to cut part B. Plant B is inserted and using the enzyme. Excellent, ligase enzyme that will fuse the two parts together. And this will port C, which is the recombinant DNA. So this is the direct question also. Part C is put into a bacterium. The bacterium is taken up by the rice plant cells, giving them the ability to produce beta carotene. Tissue culture is a form of which type of reproduction? Asexual. Again, we took it when we studied plant reproduction. We took asexual tissue culture, cuttings, and the potato tuber, but of course you will forgot. Apart from the structural features, state 2 reasons why bacteria are used for genetic modification. Rapid reproduction, they're cheaper, no ethical concerns over their use. They have a plasmid, which is used as a vector to transport genetic material. They have a universal genetic code like all the other species. There's so many reasons why we use bacteria. Can you write the disadvantages of genetically modified rice plants? In other words, what are the disadvantages of genetic? Yes, I want 3. More or less? Less variation. You said what? What do you mean more diseases? No, you need to be more specific. In what? Yes, you're close. Yes, 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 yes. Okay. Also expensive or less expensive? Expensive. It's not, it's harmful. It can cause allergic reactions. Here, less variation. And there is a chance that you can create bacterial strains that are harmful. This is what you were trying to say. Yes, it's not ethically acceptable. Yes. Also, we can create herbs that are resistant to herbicides or pests that are resistant to pesticides. Yes. Where is it written? 1 minute, I'll tell you now. It's written here. So we'll take the list as a whole. Again, all this is supplement. Why? Because this is a clear objective that says the advantages and disadvantages of genetically modified crops. So you will write the disadvantages here. Expensive, harmful, can cause allergy, can cause cancer. They reduce biodiversity. When they reduce biodiversity, plants and animals can become extinct. They can create harmful bacteria by accident. Not ethically acceptable. Resistance will develop. Habiba, are you writing the answers? Okay, very good. You have it. Very good. Please get the past papers. I mean, you don't have to purchase them. Download them. They just have to be a hard copy. You have to practice writing. This is not going to work. Okay? Or if you have an iPad and you can write on the screen, but it's not very, it's very annoying, believe me. It's very good if you write with your own hands. Done? Is the exam finished? Okay, scurvy is because we have lack of which vitamin? Very good, rickets. Perfect. We're done. So this exam, in my opinion, is very easy. Most of the questions are direct, straightforward, and a few tricky questions. You study well, paper four, study year 10 notes. Okay? Year 10 notes will ace you. But everything, because it's short and summarized and to the point. It doesn't waste any time. Objective by objective. And from the beginning till the end. Year 10 done and you have time, do 9. Okay? Very good. On Saturday, we have revision. I'll revise inheritance.